everyone. Can you all hear me? Okay. So this summer, I did my research at the University of Hong Kong under the supervision of Professor Yu Wang and Dr. Daniels Kanja. And my research topic was the protective role of endothelial SIRT1 against vascular aging and hypertension. It's a mouthful, so let's break it down. Hypertension is when blood pressure is too high, and it could lead to other heart diseases like stroke. It can be caused by various factors like obesity, genetics, alcohol, but my focus is on hypertension resulting from aging. So blood flows by the contraction and relaxation of blood vessels. And when we age, our blood vessels harden and they cannot dilate properly after contracting, which will make the heart have to pump harder to allow blood flow, which leads to higher blood pressure. The vascular endothelium is the inner lining of blood vessels that is responsible for blood vessel function and blood pressure regulation. And SIRT1 is a gene that is responsible for the whole body energy balance, as well as regulating the aging process. SIRT1 in the vascular endothelium has the following function. First, it can prevent any aging-associated dysfunction in the vascular endothelium by preventing the cells from losing the ability to renew and divide. It can also regulate blood vessel contraction and relaxation, which in turn regulates blood pressure. And all this together will help reduce the risk of developing cardiovascular diseases. So here are my results. To reach the objectives of my study, I looked at 3 types of mice. So first are wild-type mice, indicated in blue color in panels A, B, and C. These mice don't have any type of genetic modification. And then our EC SIRT1 mice, indicated by green, who have been genetically modified to overexpress human functional endothelial SIRT1. And the red are EC H363Y mice, who are mice with dysfunctional human endothelial SIRT1. As we can see in panel A, when SIRT1 is dysfunctional, the body weight increased significantly more over time, indicating that a lack of a functional SIRT1 reduced metabolism greatly. In B, it is clear that the mice with dysfunctional SIRT1 have way higher blood pressure, and similarly in C, their blood vessel contraction is greater, because more blood vessel contraction means higher blood pressure. And finally, in D, we are looking for those cells that have lost the ability to renew and divide. And we can see that in the mice with dysfunctional SIRT1, EC-H363Y mice, these type of cells indicated by blue stains are way more in number as compared to the other 2 type of mice. So altogether, we can conclude that when SIRT1 is present, metabolic activity increases. Vessel contraction reduces, which also reduces blood pressure, and also reduces the cells which lost the ability to renew. And altogether leads to normal, healthy blood pressure. But the complete opposite happens when SIRT1 is dysfunctional. Now that we know the protective role of endothelial SIRT1, how it can protect our blood vessels in such ways, we can look into the therapeutics potential of SIRT1 and how we can utilize this gene to help us overcome the challenges of aging, which is hypertension. Thank you all for listening, and thank you to all that helped along the way to make this research possible.